Male reproductive system. Structure of the male reproductive organ. The male reproductive system consists of primary sex organ, which is testis, and accessory sex organs like epididymis, vas deferens, seminal vesicles, prostate, and penis. Testis. The testes are paired oval structures, each weighing about 10 to 15 grams, 5 cm in length and 2.5 cm in width, and are located outside the abdominal cavity in the scrotum. The temperature in the testis is generally 2 degrees lower than the body temperature, which is ideal for the optimal development of the sperm. The testis in males relate to ovaries in females. The human testis is covered by a thick connective tissue capsule which is called tunica albuginea and is divided into numerous lobules by the fibrous septa. Each lobule encloses 3-4 loops of seminiferous tubule. The production of sperms occurs in seminiferous tubule. The seminiferous tubule has a basement membrane which separates it from the Leydig cells and myoid cells in connective tissue. In addition, the seminiferous tubule contains Sertoli cells and germ cells. Sertoli cells. Sertoli cells form the major mass of the seminiferous tubule. The cells are large, irregular shaped, which extend from the basement membrane to the lumen of the seminiferous tubule. The apical membrane has sperms attached to them. Sertoli cells are attached to each other by tight junctions present at their bases. The tight junctions separate the seminiferous tubule into 2 compartments: the basal and the adluminal compartment. The basal compartment is the outer compartment which majorly consists of spermatogonium. The spermatogonium is in close contact to the blood vessels, and there is an exchange of substances. The adluminal compartment is the inner compartment of the Sertoli cells, which contains primary and secondary spermatocytes and spermatids. Thus, the tight junction separates both the compartments of the seminiferous tubule, leading to the formation of blood-testis barrier. The blood-testis barrier prevents the diffusion of large molecules into the interior of the tubule and protects the germ cells from the harmful substances present in the blood. The Sertoli cells help in the formation of blood-testis barrier, provides nutrition to the developing sperm, and destroys the defective sperm. In addition, it also helps in the production of inhibin, estradiol, androgen binding protein, and Mullerian regression factor. Germ cells. Germ cells, or spermatogonium, are the undifferentiated germinal epithelial cells of the seminiferous tubule, which are present near the basal lamina of the seminiferous tubule, and they develop into spermatocytes and spermatozoa in the process of its development or spermatogenesis. Interstitial cells of Leydig. Leydig cells are the cells which are present in the interstitial space between the seminiferous tubule. The cells are rich in mitochondria and endoplasmic reticulum. The cells mainly secrete testosterone. The Leydig cells are active at 8 to 12 weeks of intrauterine life for the differentiation of the male genital ducts, and they remain dormant till puberty. On the onset of puberty, their activity is enhanced, and thereafter, their activity decreases. Each loop of seminiferous tubule empties into the anastomosing network of tubules, which is called the rete testis. The rete testis further continues the small ducts called the efferent ductules. It is through this sperm leaves the testis into the head of the epididymis. Accessory sex organs. Epididymis is the highly coiled tube and is 6 meters in length. The epididymis is divided into 3 portions, which are the head portion, which is close to the testis, body, and tail portions. Epididymis stores the spermatozoa and aids in its maturation. Secretions of the epididymis provide nourishment to the spermatozoa. Once the sperm passes through the epididymis, it attains complete motility. The tail portion of the epididymis continues as the vas deferens. The contraction of the walls of the vas deferens helps in the movement of the sperms. The seminal vesicles are lobulated glands which lie close to the prostate. They secrete thick viscid fluid, which is alkaline in nature. The fluid makes up to 70% of the semen volume. The seminal vesicle joins the vas deferens to form ejaculatory duct. The ejaculatory duct acts as a storehouse for the amount of semen that is ejaculated during intercourse. The prostate is the largest gland of the male reproductive system. The urethra passes through the center of the prostate and joins the ejaculatory duct in the prostate. The prostatic fluid is rich in fibrinolysin, fibrinogenase, and acid phosphatase. The penis is a copulatory organ of the male and is the common opening for urine and semen. Penis has 3 erectile tissues, which are the right and the left corpus cavernosum and corpus spongiosum. The vascular structure, which is located at the tip of the penis, is called the glans and is identical to the clitoris in female. Spermatogenesis. Spermatogenesis is the process of development of sperms from the germ cells. The primordial spermatogonia divide mitotically to form daughter spermatogonia. Few of these daughter spermatogonia migrate apically from the basal lamina and enter into the first meiotic division, and these cells are called the primary spermatocytes. On completion of first meiotic division, the diploid primary spermatocytes give rise to haploid secondary spermatocytes, which again undergo second meiotic division. Haploid set of spermatids are produced at the end of second meiotic division. Spermatids are small rounded cells that undergo series of metamorphosis to produce mature spermatozoa, and the process is called spermiogenesis. Spermiogenesis. During the process of spermiogenesis, the size of nucleus decreases. An acrosomal cap is formed on the sperm head, which contains hydrolytic enzymes that are necessary during fertilization. Along with the formation of the middle and the tail piece, which helps in the swift motility of the sperm. Most of the cytoplasm is lost as residual bodies as the sperm matures. Spermatozoa remains attached to the apical surface, the seminiferous tubule. The release of spermatozoa is controlled by Sertoli cells. The process of release of spermatozoa by the Sertoli cells is called spermiation. The process of formation of the mature sperm from the germinal cells takes about 60 to 70 days. Factors affecting spermatogenesis. Luteinizing hormone, which is secreted by the anterior pituitary, stimulates the Leydig cells to secrete testosterone, which increases the formation of sperms. Similarly, follicle-stimulating hormone secreted by the anterior pituitary also facilitates the maturation of the sperm and also initiates the spermatogenesis at puberty. Estrogen, which is secreted by the Sertoli cells, aids for spermiogenesis. Growth hormones are necessary for the normal functioning of the testis and also division of the spermatogonia. In addition, temperature of the testis is also important for the spermatogenesis. 32 degrees centigrade is the ideal temperature for the development of the sperms, which is maintained by a scrotal sac that encloses the testis. Exposure to harmful radiations like X-rays bring down the normal production of sperms. Structure of the mature sperm. Sperm measures about 55 to 65 micrometer and moves at a speed of 1 to 4 millimeter per minute. Mature sperm is composed of 3 portions: head, neck, middle piece, and the head is oval in shape and is 4 microns in length and contains condensed nucleus. The anterior portion of the head is covered by acrosomal cap, which contains hyaluronidase, acid phosphatase, and mucopolysaccharide enzyme, which helps in the penetration of sperm into the ovum. The neck is a small constriction between the head and the middle piece that measures about 0.3 microns in length. At the posterior pole of the nucleus, 2 centrioles are present, which are proximal and distal centrioles. The middle piece is long, cylindrical in shape and made up of axial filaments and is surrounded by mitochondria, which provide energy for the movement of the sperm. The tail piece or principal piece is the motile part of the cell. It is the longest part that measures about 40 microns long and 0.5 microns in diameter. It contains from within outwards axial filaments, fibrous sheath, thin cytoplasm, and cell membrane. Semen and its composition. The fluid which is ejaculated from the male genital tract during orgasm is called semen. Normal volume of semen which is ejaculated during one intercourse is about 2 to 6 milliliter and has a sperm count of about 60 to 120 million per milliliter. It is milky white in color and has a pH of 7.4 to 7.5. The semen contains 10% of sperm and fluid from vas deferens, 60% of seminal fluid, and 30% of fluid from the prostate. Applied aspects. Oligospermia. It is a condition in which sperm count is less than 15 million per milliliter. If the sperm count is between 10 to 15 millions per milliliter, then the condition is mild oligospermia. If the sperm count is between 5 to 10 million per milliliter, then the condition is called moderate oligospermia. And if the sperm count is below 5 million per milliliter, then the condition is called severe oligospermia. Azoospermia. Azoospermia is a medical condition in which there is a complete absence of sperm in the semen ejaculated during orgasm. This condition causes infertility in males. Abnormal morphology of sperms. Abnormal sperms include sperms with bifid head, bifurcated tail, an abnormal shape of the sperm, and absence of sperm head. These abnormal sperms are infertile and do not have the capacity of fertilizing the ovum. Physiology of male sexual act. The sexual act in male occurs in 3 stages, which are as follows: erection, emission, ejaculation. Erection. Erection of penis occurs due to dilation of arteries, making the erectile tissues of penis corpora cavernosa and corpus spongiosum being stimulated by parasympathetic nerves, nervi erigentes. Acetylcholine, vasoactive intestinal peptide, and nitric oxide are released, which causes increased blood flow and filling of the erectile tissues. All these make penis hard and elongated. Emission. In this stage, the semen moves into the urethra due to the contraction of the smooth muscles present in the seminal vesicle and vas deferens. Ejaculation. This stage occurs during orgasm, where the semen moves out of the urethra due to the contraction of the bulbocavernosus muscle. Testicular hormones. Testes secretes male sex hormones which are together known as androgens. Androgens secreted by the testis includes testosterone, dihydrotestosterone, androstenedione. Testosterone is the male sex hormone which is synthesized in large quantities. Yet, dihydrotestosterone is more active. Testosterone is secreted by the interstitial cells of Leydig, which are present in the seminiferous tubule. These interstitial cells of Leydig constitute about 20% mass of the adult testis. 4 to 9 milligrams of testosterone is secreted by the testis every day. However, a small amount is also secreted by the female ovaries and adrenal glands. Testosterone is a C19 steroid hormone with OH group present at the 17th carbon atom. Both cholesterol and acetyl coenzyme A is the precursor of the synthesis of testosterone. 98% of the testosterone is transported in bound form with the plasma protein, of which 33% is transported as plasma albumin and 65% is transported as testosterone-binding globulin. Only 2% of testosterone is transported as free form. In many target tissues, testosterone is converted into dihydrotestosterone, which is the most active form of androgens. Testosterone is converted into estradiol in tissues of adipose, hypothalamus and liver. Major portion of testosterone is degraded in the cells of the liver, where it is converted into androsterone and dehydroepiandrosterone, which are excreted through bile or urine. The secretion of the testosterone begins at 7th week of fetal life, and the fetal testis begins to secrete testosterone at about 2nd to 4th month of fetal life. In childhood, there is no secretion of testosterone, and the secretion of testosterone begins at the onset of puberty. However, there is reduction in the secretion of the testosterone at 40 years, which is called andropause, and the secretion becomes almost zero at 90 years. Functions of testosterone. Testosterone is mainly responsible for the masculine characters. In fetal life, it is prominent for sexual differentiation of the fetus. During the 7th week of intrauterine life, there is production of testosterone. Before 7th week, the fetus will have both Wolffian and Mullerian ducts. Male sex chromosome will cause the genital ridge to secrete testosterone, which results in the development of male accessory sex organs. In case there is no testosterone production, the development of Mullerian duct continues, and accessory female sex organs are produced. In addition, testosterone also is necessary for development of accessory sex organs, such as the scrotum, penis, genital ducts, prostate, and seminal vesicles. Testosterone helps in the descent of the testes from the abdominal cavity into the scrotum. During onset of puberty, testosterone increases the size of penis, scrotum, and testes, and also helps for spermatogenesis in males. Testosterone is very much necessary in development of secondary sexual characters in males, like effect on muscular growth, growth of bones, development of hairs in pubic, chest, limbs, back, face, and other parts of the body is under the influence of the testosterone. Testosterone influences cracking of voice due to enlargement of larynx.